Hey everybody, Mr. Rogers here, and we're going to do our last note for Punnett squares. And what we're really looking for here is we've done things like this. We've done Punnett squares for this whole week. We've actually done Punnett squares that kind of cover the same ideas we're going to do today, but you really have to pay attention to the story problems with these guys, okay? So the first one we're going to talk about is multiple alleles. Okay, multiple alleles. And multiple alleles, it means that there are two or more variations. I guess it should be three or more, technically. Three or more variations that could exist. And you actually did a multiple allele problem yesterday, and that's blood typing. Human blood typing is multiple allele because there was IA, IB, and I. There was three variations, and how you combine these created different genotypes and phenotypes. Real life kind of looks more in multiple alleles than it does in what the standard genetics is for humans. And to give you an idea, this isn't exactly right, but it's heading down the right path. Let's do like multiple allele question with human hair. So we got multi alleles. So we got multi here. So we're going to do FA equals brown hair in humans. FB, again, F is just really like a standalone. It just means technically whatever you want it to mean. I picked F randomly because when I was first starting this question, I was going to do fur. I think that's why, and then I switched it to human hair. FC equals blonde hair. And FD equals red hair. Now, using this sometimes multiple allele, they'll tell you where the hierarchy of dominance means like which one is more dominant than all the other ones. And so for this question, brown hair is the most dominant, and red hair is less, and it goes down. Okay, and if I have both. If I have two alleles, well, I'm going to have two alleles, but if I have two different alleles, it's codominant. So they both show. Okay. So this is kind of more or less how we get hair color. We start getting hair color this way. And truly, by the way, you could say codominance, but we can also say incomplete dominance. What I mean by this is if you actually microscopically look at someone's hair, it is technically more codominant. In the hair, there will be streaks of different pigments. But when you look at it from a distance, it looks incomplete dominance. Like you can't tell if someone had brown hair and blonde hair. There's like, you can't tell from even like 1 inch away that there's streaks of different colors, but there are. So it'd be codom, but we'll let it go. Anyways. So we'll say somebody that is F big A, FC, so brown and blonde, crosses with someone that is FD, FD. The only way in this question, and they'll tell you these things, to get red hair is to be FD, FD. So this person would have straight red hair right here. This mom does. The dad is a mixture of brown and blonde. So we go through. And we find out that it's going to be multiple different things here. Really not that much. I mean, it's 50% this, 50% this. So this child, their children, could be 50% FA, FD, which would be brown red, so like a brownish red, and 50% would be a strawberry blonde because it'd be red blonde. Okay, and so this is how this works. And so there's not just two alleles; it's going to be multiple alleles, and they're going to interact with each other. That's a multiple allele. What epigenetics is, or epistasis, I should say genetics, just epistasis, is one trait affects another one. And so when you read those questions, I'm not going to go through an example of it, but it will say something like, if you have somebody that's A, big B, or A, little a, big B crossed with big A, big B, big B, it will say something along the lines that if someone has a homozygous recessive little a, then it changes everything. So you got to read the question, okay? I'm not going to go through an example of this, but make sure you read it and understand like what happens if all these different things happen. So almost think of epistasis almost like light switches. So when I grew up, I had a light switch over by my doorway, and I had a light switch over by my bed. And if this light switch was off, no matter if I turned this one on or off, the light would stay off. This one had to be on for me to be able to turn on and off over here. And so that's kind of epistasis works, is one of these guys affects the other one and how they work. When we talked about dihybrids earlier this week, I made a real big focus on it being an independent gene because epistasis are dependent genes. These depend on each other. So they're going to be dependent on each other. If something happens here, it affects the other one over there. All right, one last thing. Gene mapping is completely different. This is not actually a Punnett square. This is just something we kind of have to learn. Gene mapping is talking about where on a chromosome a gene lies. Okay, where on the chromosome the gene lies. And how we do that is we talk about how much separation there is between these different genes. If the separation is greater than 50%, if it's greater than 50%, we say it's really far away. If it's less than 50%, then it's close and we need to be careful with it, okay? Now, what these percentages mean is the likelihood of crossing over in meiosis. Okay, likelihood of crossing over in meiosis. We talked about meiosis last week. So in prophase 1, between prophase 1 and metaphase 1, there is a crossing over event where these guys are going to be switching arms and legs, these chromosomes, before they split up into different haploids. This is a scientific way of saying how close something is. So for example, if something is here, say this is gene A, and this is gene B. Sorry, my lights went off. I'm trying to get them back on. This is gene B, and they are 25% apart from each other, or 25 away from each other. It means 25% of the time, a crossing over event could separate these two. That the cut could be here, here, anywhere in there. Okay? If B and C are right next to each other, and we say that this difference is only 2%, we're saying there's a very low chance that these two could be separated from each other. That these two are very close. C and D, if I put D all the way over here, and this is 40% between these two, there's a 40% chance C and D will be separated if we did a crossing over event somewhere in the chromosome. So it lets us know what's the percentage one of these guys being split up and maybe going to different chromosomes in the crossing over event, but also it tells us how close they are relatively. The closer they are, the more likely that they are going to stay with each other. We can also use this to kind of figure out where other things are. Like I never told you how far away A and D were, but if I add everything up, 40 to 25, now we're at 67%, which is greater than 50%, which means they're really far away from each other. Okay. Anything over 50%, we just automatically say really far away. just so you guys know. I mean, that's still probably right down 67%. But if they ask me how close are they together, I would say really far away. Usually, if they're that far away from each other, they actually exist not on the same chromosome, but different chromosomes. And that's a whole different story. We're not going to get into that right now. So G mapping, they're going to tell us where different things are. And we kind of have to map them out. We have to do some logic. It's kind of like a puzzle. And we say what these actually mean. 25% means what's the likelihood of crossing over happening between those two genes. Okay? So those are the three things we're talking about today. You're going to do some problems. They're two different worksheets. One has 3 questions. One has like 3, I think, too. I think they both have 3. Work through them, and if you need any help, I'm here. We're going to go through them again on Monday or Tuesday. These are the hardest ones, so if you need help, please come and get help. All right, guys. I'll talk to you later. Bye.